Okay, this is what the anther and the connective portion, that is we call them as a filament. So when you just see the powdery mass which is present in the form of anther, the agglomerated form of pollen grains, we call them as anther. This powdery mass, when you observe it under this scanning electron microscope, I think everyone is familiar with the scanning electron microscope. In the previous semester, they have gone through the different types of the microscopy, transmission electron microscope, scanning electron microscope, and compound microscope, and there are different type of electron microscopes, right? So when we have observed this powdery mass in the scanning electron microscope, you could able to see the structure like this. Okay, now so just on out of curiosity, would like to tell you that this is what the structure of the pollen grain of the marigold. Have you heard the name of plant marigold?
Yes, sir.
Okay, now this is what this pollen grain. So can you tell me which is this? This is also again a different, a beautiful structure of the pollen grain. So can anyone guess me? Have you ever seen such pollen grain? There are, when you go in detail about the pollen grains, the scanning electron microscope scanning electron microscopy images of the different pollen grains. You just Google it when you find free time. So just Google it, you will come to know the different structures of the pollen grains. So this is what the pollen grain of rose, Rosa chinensis. Okay, na? So these today why I'm addressing only towards the anther and the pollen and the filament is that, so during the course of plant breeding or production of the haploids, production of the monoploids, production of polyploids, the the different scientific community has started to work on isolation or identification or selection of the haploid cells. So usually where you are going to get the haploid cells, either in the male reproductive structure and or in the female reproductive structures. Okay, now, so as per the demand of the syllabus, we are supposed to study the haploid plants or a production of haploid plants using anther and the pollen. Okay, now, so there is one more point, that is you can even study by inoculating the gynoecium part. Okay, na? So the female reproductive part you can take and you can go for production of a female plant. So if you are taken the anther or a pollen grain, you are going to get the microspores, which results in the production of only the male plant. Why? Because these anther or a pollen are the male reproductive structures. These contains, they after these microspores are forms by undergoing the meiotic divisions. So what happens during the meiotic division? This is what we commonly call them as reductional cell division, right? So this reductional cell division, which are made diploid cell into two haploid gametic cells. Okay, now so one haploid cell will be the male reproductive structure and another haploid cell will be the female reproductive structure. Are you getting my point? This is what the common thing what we have studied in the previous semester, that is in the embryology, right?
Yes, sir.
Okay, so next coming to the, what do you mean by, you need to study what do you mean by haploids, what do you mean by haploid cultures. Coming to the haploid plants, usually the haploid plants stands for, or which are usually characterized by having only single set of chromosomes. Okay, now see these single set of chromosomes usually derived from the gametophytic numbers. So that's what beginning of the class I had explained you all that. The usually diploid number of chromosomes, which is nothing but they contains the somatic chromosomes as well as the reproductive chromosomes. Okay, now, but when it comes to the haploid cells or haploid plants, they contain only single set of the somatic chromosomes. For example, if the plant original or a homozygous or heterozygous plant comprising 20 chromosomes, then the haploid plant will contain only the 10 chromosomes. The another haploid plant will contain the 10 chromosomes. Okay, now, so where, so this particular haploid which resembling the male characteristic, then it can be said as haploid male plant. So if this particular plant which comprising only 10 chromosomes female flower, then it is said as a female haploid plant. Okay, now nothing but this plant may produce the flower but that particular flower comprise only the male reproductive structure, that is only the androecium, only the anthers. And this particular plant which is going to produce the flower, but the flower comprising only the gynoecium part, that is what we call them as a pistil or a carpel, or which is a unit of style, stigma, and ovary. Okay, now, so what do you mean by this? Gametophytic number of chromosomes will be there in a complete plant. So when you go and start observing the each and every cells, so you will get each and every cell comprising only uniform number of half of the or a gametophytic number of the chromosomes. Okay, now, so why this haploid culture has came into existence? I mean, why the haploid cultures were needed for the plant tissue culturist, or why the haploid culture concept was begin and when? Okay, now, so the main reason or principle behind the haploid culture is that these haploid plants, which are having a greater significance in production of homozygous lines. For example, let me tell you that, so if you have a plant which you don't know whether it is a homozygous one or a heterozygous one. Okay, now, so for example, you have taken a heterozygous plant, but in plant breeding, heterozygous plant, if we undergo the crossing, then their offsprings may be possess the dominant characteristics, or it may possess the recessive characteristic because of a heterozygous characteristic, right? So there, if you want to produce homozygous characteristic, let us take, we have a plant, capital A and small a. It's heterozygous in condition. Okay, now, so when you go on for production of haploid plant, one plant will comprise only capital A, the another plant will contain only the small a. Nothing but we have produced two haploid plants. Okay, now, so this particular plant, by treating different chemicals or by radiation influence, we can make this particular capital A, so we can double the chromosomes by different agents. So we'll explain what are those agents in my coming classes later on. But this particular cell which comprising only the A characteristic, nothing but half of the chromosomes, pardon, half of the number of the chromosomes, then we can double them by treating different chemicals. The best example for the chemical which is being used for the doubling the chromosome, that is the colchicine. Okay, now produced by the haploid plants. Okay, now, and not only that, the for the first time haploid existence, the haploid existence for the first time discovered by Bergner in the year 1921, which is in the Datura starmonium. Okay, now, so in the Datura starmonium, which is, can be said as the first haploid, not the first, the existence of the haploid from the Datura starmonium was reported by the Bergner in the year 1921. Later in the year 1964, Guha and Maheshwari, these two are the two pioneer embryologists of India. Okay, na? Guha and Maheshwari, both are male. Don't get confused with the Maheshwari as a female name. Panchanan Maheshwari is a full name of the person. He is a well-known, a renowned embryologist. Okay, na? And they have reported, okay, na? They have successfully reported the haploid embryo formation. Later on, those embryos which are going to produce or form the plantlets, okay, now, these embryos, that's what we call them as a somatic embryos, right? In the laboratory or in vitro condition, where the embryos which are forming, because these embryos are forming, later these embryos are converting, or because of the bipolar development, they are forming the plantlet. Nothing but the shoot is also forming and the root is also forming. Right, this particular embryo and the plantlet formation have been derived from the microspores. So what are these microspores? Where you are going to see the macro and microspores. Microsporangia structure in your mind, you will come to know what are in the four corners. You are going to see the four pockets. There you could see the microspore, which are in present initially in a round form. Later, as they develop, they become four in shape, tetrad condition. Okay, now that particular tetrad condition, which is formed by the microspore mother cell, commonly called as MMC, abbreviated as microspore mother cell. Later, it has become microspore, and these microspore, which are are haploid cells. Even the same thing happens in the megasporangium. There we are going to see the megaspores. Usually these microspores are nourished by the tapetum, right? This tapetum, which is a nutritional layer which is present in the innermost wall of this microsporangia. In the year 1961, Borgin and Hitch in the year 1967, he has a full-fledged information or a report or a composition for the nutrient required for production of a haploid plant from the Nicotiana tabacum has been reported. Nothing but, so if you follow Bourgin and Hirsch, for the production of a haploid plant from Nicotiana tabacum, you can just refer the report which have been presented in the year, reported in the year 1967 by Bourgin and Hitch. Okay, he just given a complete composition of the nutrient medium and at what stage the microspore has to be taken and how to inoculate them and what is the time light and dark condition to be given, what is the light intensity to be supplied and how to maintain the cultures, what is the temperature needed for that, what is the humidity condition is required, everything with regard to the Nicotiana tabacum. He has not given any such information about other plants, but we can refer for other plants too. Okay, now, but a standardized protocol for obtaining the Nicotiana tabacum haploid plants was been given by Bourgin and Hirsch in the year 1967. So be achieved by two means. Okay, now one is in vivo methods and the second one is in vitro methods. What do you mean by in vivo methods? What do you mean by in vitro? In vivo and these in vitro are these two are the Latin words. So just whenever writing these two in vivo or in vitro, you need to italicize that. Okay, na? Okay, when you are typing those. And the next point is that this in vivo stands for when you are studying or in the methodology applied naturally within or with the plant, that particular condition or a technique or a mechanism or a phenomenon, we call them as in vivo. Then in vitro stands for that's completely an artificial medium that's on in the laboratory, that is in the glasswares, in the glass vials, with by providing all the artificial environmental condition. Now coming to the in vivo techniques, what are the techniques which have been adapted or which are employed for production of haploids? So the first one we have androgenesis, gynogenesis, distant hybridization, irradiation effects, chemical treatments. So by these things, in vivo, I'm not talking about the in vitro, okay? Na? We in vivo, within the plants or within the plants, we can, by using all these 5 techniques, by applying all the 5 techniques, any one or all the 5 or any 2, any 4, any 5 or any 4. So you can employ and you can go for production of haploid plants. For when you take the androecium or a male reproductive structure for culturing and obtaining the haploid plant. Okay, now that's what we call them as androgenesis. Okay, nothing but production of a haploid plant which is dominated by the androecium or a male reproductive structure. Nothing but production of a only male reproductive organ or a male flower containing plant. Gynogenesis stands for again production of a plant which is comprising only the female reproductive structure. It doesn't mean the stamen should not be there. There are sometimes during the gynogenesis or sometimes when you in a plant to be said as female haploid plant, there sometimes the stamens may be there, but those stamens which are infertile, they contain the staminodes. So at even that condition also we can claim that as female haploid plants. Sometimes these stamens or staminodes may not be there. Only the flower comprising only the female reproductive structure. So these two possibilities there where in vivo we can go for production of the haploid plant. Now coming to the distant hybridization. So what happens? Distant hybridization during crossing when we had, as I already told you that when we want to go for production of a homozygous plant. So, but because even after repeated crossing and it maybe anything, the homozygosity is not possible in that particular plant, then we can go for distant hybridization, nothing but between the two genera, or between the two families for distance, the plant which is nowhere really interconnected between the variety, between the species. There must be hybridization between the two different genera, two different families. There what happens usually, we can go for production of that and we can nullify the expression of one plant, one genera or one family where which is not being needed for production of that characteristics. Okay, now, and that's we can enable only through the plant breeding or hybridization techniques. So at that time, at that time, what happens? One particular genera or one particular family, let us take it as a bee. A and B, when we are hybridizing two different genera or two different families, where we are supposed to get a plant comprising A, B characteristics, right? But because of some external factors, we can nullify the expression of B characteristic. Nothing but what happens, the particular plant which comprising only the A characteristics can be produced by distant hybridization. Okay, now, so anything can be possible due with distant hybridization, but prior to that, you must have, or a person one who is working with the plant breeding has to know the possibility of the crossing, and also one must have an idea of how the crossing is being done. It should not be in such a way that instead of expression of only A characteristic, there should not be any B characteristic. You must, even the person on the plant breeder must have the idea of the what we call the information about the genetic genotypic information of those two plants. Okay, now, so through the distant hybridization also there is a chance or a possibility of production of haploid plant. Next, coming to the irradiation effects. Irradiation effects in that by exposing some of the plants to the radiations, so we can make a plant to a monoploid. Okay, even I think I had forget to explain that. We have haploid and diploid. Haploid means a plants which comprising only single set of chromosomes. Diploid stands for usually a plant which comprising two set of chromosomes. Okay, so what do you mean by monoploid? In haploids, we can broadly we can classify them into two types. One is monoploid, another one is monopolyploidy. Okay, now what do you mean by monopolyploidy or monoploidy? So monoploidy, for example, if a particular plant which is having 2n set of chromosome, then their haploid cell must contain only single set of chromosome or 1n set of chromosome. That condition we call them as monohaploid or a monoploid. And polyhaploid, here we have a particular, for example, in a plant which is having more than 2n set. Sometimes it may be having 4n, 5n, 10n. Okay, now, that particular haploid in this polyploidy condition, their offsprings or their gametic cells contains half of the chromosomes. So, for example, a particular plant is having 10 set of chromosomes. Their gametic cell will be containing 5n set of chromosomes. Right? At this such condition, we call them as polyhaploids. Okay, now monohaploids are only the cells or gametic cells or plants which comprising only single set of chromosomes, where the polyhaploid stands for the plants which are comprising the of the chromosomes of the polyploidy parent or a polyploidy plant. And irradiation effects, here there are some UV or IR radiations can enable the what we can make or we can artificially kill some reproductive structures. Or if you want to go for production of if you want to have only the male plant or androecium containing plant, then we can expose the gynoecium part to the UV or IR radiation, so that what happens, these two which will cause major adverse effect. Finally, these two or some other radiations may happens to kill, or they may destroy the effect of they may destroy the gynoecium activity. Okay, now, so what happens, only the male reproductive structure will be there in the flower and a particular plant, then we can say that particular plant is a haploid plant. Now coming to the chemical treatment, and there are different type of chemicals can be treated. So for when a flower which has been exposed to the chemicals, there are different chemicals. We'll explain what are those chemicals which can be used for production of only haploid plants. Though a particular plant is only said as diploid, by treating the particular plant with the suitable chemicals, we can make those particular plant as an haploid. Nothing but what do you mean? It doesn't mean that the complete plant is going to be formed only with the male flower, or a complete plant is going to be formed with the female flower by these two irradiation or chemical treatments. Okay, so here these two are acting as an agents for killing or destroying the effect or masking the impressions of the female or a male as per the requirement of the person who is going to work on that. Now coming to the in vitro techniques. Now we'll so that's we have studied with or within the plant body. That is what we call them as the in vivo techniques for the production of haploid plant. Now coming to the in vitro techniques which have been employed for production of haploid plants. Here we have the androgenesis and the gynogenesis. Only these two possibility, nothing but if you want to look for male haploid plant, we can go for production of you can depend on androgenesis, and if you want to produce a female haploid plant, then you have to depend on gynogenesis. There is only two possibility in vitro. What happens in vivo, already the plant is already available. So by using distant hybridization or by irradiation influence or by chemical influence, we can make the plant to haploid. But because already the mother plant is available, we can manage the diploid plant into haploid plant. But in artificial or in vitro techniques, we don't have any starting material. We don't have any plant already. There you need to go for selection of the pollen grain or anther for the androgenesis, or we can go for selection of ovary for gynogenesis. So if you are looking for a male haploid plant, then the sole dependency is on the androgenesis, and if you are looking for the female haploid plant, then you are supposed to depend on gynogenesis, right? So these two are the only two methods which can be employed through in vitro technology for the production of haploids. Nothing but selection of a male gametophyte. Okay, now so male gametophyte, nothing but the androecium containing the male gametophyte. Usually this male gametophyte or anther, when you dissect, so you are going to see the microspores. So if the flower in bud condition, or if the androecium in bud condition, or if stamen or anther in immature condition, you are going to see microspore mother cells. But when as the developmental stages we could see, there finally you are going to see a mature microspores. These can be said as the pollen grains. Okay, na? So these male gametophytic or microspores or pollens, usually immature pollens need to be taken for culture. Okay, na? So these produces male or haploid plant. Okay, now nothing but a plant which comprising single set of chromosomes of the original plant. As already told you that usually onion contains 12 to 16 number of the chromosomes. So during the meiosis study, you will find only 8 to 12, pardon, 6 to 8 number of chromosomes. What is the main principle behind the androgenesis? The main principle is that to stop the development of pollen cell into gamete. Okay, instead of usually what happens during the fertilization, you would study when the pollen tube reaches the ovary, what happens during the development occurs. Well, the female and the two male gametes are going to form from the pollen, right? So one will fuse with the egg cell, another one fuse with the nuclear polar nuclei, right? Here the main principle behind the androgenesis is that instead of making a pollen cell divide into two male gametes, we are artificially asking or forcing the pollen cell to develop into a complete plant. Since the pollen contains only the single set of chromosomes, henceforth the plants which have been produced by from that particular pollen must contain or will be containing the single set of chromosomes. Those we call them as haploid plants. Okay, now, so this haploid plant production through androgenesis can be done by two methods in vitro through in vitro techniques. One is anther culture, another one is a pollen culture or microspore culture. Okay, now, so don't get confused with the MS stands for here. This particular here microspore. Okay, now, not Murashig and Scoob medium. Okay, now, got it? So these two are having their own applications, advantages, and their own limitations, and those will explain in my next coming class.